Hello, my name is Nicole Hansed, and I am a third-year Bachelor of Biological Sciences student majoring in biochemistry, and I am excited to discuss my summer research entitled Antimicrobial Activity of Endolysins and Membrane Destabilizing Agents on Mannheimia haemolytica. Bovine respiratory disease is one of the leading causes of morbidity and mortality in Canadian feedlot cattle, causing significant economic losses in the beef and dairy industry. The primary bacterial agent responsible for this disease is M. haemolytica, and it has been discovered that all strains of this bacteria are resistant to at least one antimicrobial class, causing the treatment of bovine respiratory disease to become increasingly ineffective. Endolysins are enzymes produced in the final stages of the lytic cycle of bacteriophages and have the potential to inhibit bacterial growth. They inject their viral genome into a bacterium within their specific host range, replicate, and cause cell death through the degradation of the peptidoglycan layer upon progeny release. The use of bacteriophage-derived endolysins has been successful in the treatment of multiple gram-positive bacteria, but the treatment of gram-negative bacteria, such as M. haemolytica, has been proven to be more difficult due to the presence of an additional outer membrane. Therefore, in order to generate an effective treatment, an additional membrane destabilizing agent is required to permeate the outer membrane and allow the endolysins access to the peptidoglycan layer. In this study, we hypothesized that the combined use of endolysins and membrane destabilizing agents to disrupt the outer membrane of the cells would be bacteriostatic towards pathogenic isolates of M. haemolytica. Our objectives were to determine the antimicrobial activity of engineered endolysins 3,2 and P2 and EDTA on multidrug resistant M. haemolytica strains 587 and 330. In this study, endolysins 3,2 and P2 were derived from M. haemolytica strain 185. Endolysins 3,2 and P2 were cloned in pBAD plasmid and expressed in E. coli BL21. They were then expressed, extracted, and purified. An antimicrobial assay was carried out as follows. Overnight culture was prepared by inoculating M. haemolytica colonies in BHI broth overnight in a 5% CO2 incubator. The overnight culture was then diluted in BHI broth and incubated until it reached mid-exponential growth, or the optical density at 600 nanometers reached 0.6. The culture was centrifuged, the supernatant decanted, and the pellet resuspended in 20 millimolar HEPs NaOH twice. The solution was then serially diluted in 10 millimolars of HEPs. The positive and negative controls were prepared in 96 well plates and incubated for 1 hour in a 5% CO2 incubator. The treatment groups were composed of endolysins and HEPs, which is a buffer to stabilize pH, EDTA, a membrane destabilizing factor, or PBS. A sample from each treatment group was serially diluted in PBS, and the 10 to the negative 2, 3, and 4 dilutions were plated on 5% sheep's blood TSA plates. The plates were incubated overnight in a 5% CO2 incubator, then the colony-forming units were counted and recorded. The mathematical formula shown below was used to quantify the results. A high log value indicates the treatment was more successful in inhibiting bacterial growth, whereas a low log value indicates less success. Untreated cells represent the M. haemolytica cells treated with PBS only, and treated cells represent M. haemolytica cells combined with endolysin 3,2 or P2 and HEPs EDTA or PBS. The PBS treatment group showed high log values against the MH330 strain and low log values against the 587 strain. The HEPs treatment group showed a mixture of results. Both P2 and 3,2 had high log values against 330, however, P2 showed low log values and 3,2 showed high log values against 587. In combination with EDTA, the endolysins were successful in completely inhibiting bacterial growth of both M. haemolytica strains, as no colonies formed on the TSA plates. It was found that the combined use of EDTA, a membrane destabilizing agent, and bacteriophage-derived endolysins P2 or 3,2 were successful in inhibiting the growth of multidrug-resistant M. haemolytica strains 330 and 587. This means that the combination has great potential to replace antibiotics in the treatment of bovine respiratory disease through lysing the pathogenic M. haemolytica bacteria. Future directions include testing of the endolysins on different multidrug-resistant M. haemolytica strains, assessment of resistance development driven by endolysin application, and challenge studies to determine the long-term effectiveness of endolysin treatment. I would like to thank my PI, Dr. New and Cedra, for their help on this project, as well as the summer undergraduate research experience for funding my research. Thank you.